So this tutorial will be more in line with the lecture 6 and lecture 7 that you did yesterday. And in the morning, in the tutorial you had in the morning, it's more of kind bioinformatic centric. So this tutorial line focuses on the agrobacterium mediated transformation in potatoes specifically, but it's more of same like for every plant. So I think we should recall like what we have learned in yesterday's tutorials. So any of you would like to add what have you learned till now? You can just say simply 1 point or 2 points. It's just a quick recall so that I can continue. Yes, and that was one aspect. Yes. Yes, single guide RNA designing was also there. Yes. Yes, we have also discussed various tools for designing single guide RNA. And what else? In the tutorial 7, you must have, Aina must have told you that how to actually design those vectors. Like I have just let you know the source from where you can get those vectors and how you can design the single guide RNA. But Aina made you download that SnapGene viewer where you can download the sequence of the vector, and she has shown you the BSA1 sites specific for that PHSC and PKSC 401 vector. So in her tutorial, yes, yes, add gene, yes, from where you can download the sequence for the vector, and you simply have to digest the vector depending on the site from where you want to add that single guide RNA, and after digestion you have to just form the oligoduplex for your designed single guide RNA, for which you have to add particular restriction sites at the 5 prime end or and at the 3 dash end. So after that you have to ligate that in the vector. So that was our whole part of designing the construct, and after that you have to transform it in bacterial system in E. coli, in any of the strain of E. coli, like DH5 alpha is there and any other strains. So that was in tutorial 8, tutorial 7 I think, and we are continuing it in tutorial 9. So here we'll be talking more about the agrobacterium mediated transformation in plants. So as I am going to be tell you about the transformation in potato, so let me just quickly tell you few points about potato. Like it belongs to the Solanaceae family, as you all know, and those of you who are working in the field of plant, and it is a tetraploid. It has a tetraploid genome which with 2n equals to 4x equals to 48. So and they are readily available, and there are also a rich source of starch, vitamin C, vitamin B6, fiber, etc. And despite this, they have a low concentration of iron, which with the concentration of 20 mg per kgs, which is comparatively low when compared to other crops. I'm just letting you know these points because I'm wanted to let you know the rationale behind selecting those genes, which we'll be covering in the next slides. So the potato also consists of secondary metabolites like alpha-solanine and alpha-chaconin, which are toxic for human consumption. So, and a known concentration of more than 200 mg per kg is considered unsafe or toxic for human consumption. So, we have taken two genes for which we have optimized this transformation in potato, for which for enhancing the iron concentration in potato, we have selected this transcription factor, that is BHLH47, and studied the iron homeostasis pathway in potato, and for the regulating the secondary metabolite concentration, we have selected this 16-dox gene in potato, and we have targeted them using this CRISPR-Cas9 approach, and successfully have knocked them down. so we'll not go much in detail about it. So okay. Okay, so is this part recognizable? Like it was in the CC top tool. So right, for STBHLH47, we have identified the potential target, and using CC top uh, we obtained a numerous amount of targets which we can select, and we have selected this target, that is target 9 according to the tool, and it has this NGG here as we are talking about the Streptococcus pyogenes. So the PAM here is NGG. So this was our 20 base pair target which we have selected, and like Aina told you yesterday, we have simply added the BSA1 site in the 5' region, and to the reverse complementary side we have added CAAA towards the 3' end. So this was for the BHLH 47 part. And for 16 docs, similarly, we have analyzed it using CCTOP, and we have also used some other tools as well. But based on this, we have selected this target, that is T8, and here the PAM is also TGG, and this was our target. And we have used, okay, let me just, okay. So I have made this presentation very plain and basic. So this is basically outline you have to follow when you will going to design your experiment for plant transformation. So the first step will be the sterilization of the explant, and the explant can be your leaf or internode or your root in in certain cases. So that all depends on the plant which you are working on. So the system proves. So the first step is sterilization of the plant. As it freezes, I don't know, we have to wait. But in sterilization, what? Okay. So for the sterilization of the plant, you will require these following things. Like you have to know the what explant source you are going to use, whether leaf, internode, or root. And after that, you'll need sterile water, autoclaved 1% bevacizine, that is the antifungal you can use to wash up the tissue, as the tissue is you have to take the tissue from any external source, and for tissue culture you have to completely sterile it before proceeding with any experiment. So, and similarly, you will need tween 70% alcohol and sodium hypochlorite as well. You can also take mercuric chloride as well, HgCl2, and of course you will need MS media. So Yes. In our case, our plant fully acclimatizes in 4 weeks. It fully matures in 4 weeks. So we have taken the 4-week-old plant as an explant source, and that will again depend on the type of plant you will be using. And after that, we will be using internodes in the case of potato. We have also used leaves as well. Uh, but we are here talking about the internodes. So we'll just simply excise the stems of the 4-week-old grown plant, and after that we will chop the stem using a sterile surgical blade, and after that the explant, that is internodes, were further excised in 2 to 4 centimeter regions between the nodes, so that it became the internode section. And after that, those internode sections were washed with autoclave water for 3 times, and after that they were treated with 70% ethanol for 1 minute. This is just simply a protocol and outline. We can also provide you with this presentation as well. So So the next steps will be wash the explants with sterile autoclave water after washing it with 70% ethanol, and after that you can give the treatment with sodium hypochlorite for 5 minutes, and after that you have to rinse the tissue like 3 times with using that sterile autoclave water. And after that, at last you have to blot dry the explants, and simply after the tissue dries, you have to place it on the MS medium, because it kills the any contaminations like bacteria and all. We have also used 1% Bevestin, which is antifungal, so it reduces the fungal contamination. Yes, we have to follow multiple steps to ensure that our explant is free from any contamination. As if you don't like, it's not any hard. It's not like you have to follow these steps. It can be more tuned as well. But if the contamination come in the later stages, it will ruin your complete experiment. So I have mentioned these few steps extra. So when you will sterile your explants, and after that you will blot dry and place it on MS medium, and it will look something like this. So here these are the internodes which are steriled and placed on the MS medium. And after that the plants will start to grow, and we have maintained like this in jars as well and in plates. So now we can directly take our explant from here. So initially you have to sterile it because the plant will be growing like in pot or from any other source you are obtaining the plant. Once you have sterilized it and maintained it in a tissue culture, you can just directly open it inside a laminar airflow, and you can just simply cut it and use it as it is. You don't have to sterile it like repeated number of times, only once you have to do it. And after that, the second step will be the inoculation of this Agrobacterium tumefaciens. So here we have taken this strain GV3101 as an example, and we have revived this strain, and the strain is harboring this PHSC401 vector with the specific guide RNA for BHLH 47 and the 16 docs. So similarly, you have to just inoculate the agrobacterium cells, and similarly you have to prepare select a single colony, and from which you have to set the primary culture. This is all just again basic stuff. So you will inoculate by selecting a single colony and inoculate it in 5 ml LB medium containing these antibiotics, as it will depend on the selection of your vector. So here this strain is also resistant to Genta and Rif, so we have also added this. So depending on the strain, different strains are resistant to different antibiotics. So, and here the bacterial selection is selection for bacterial is kanamycin. So this is the concentration which we have used. And as this agrobacterium grows in 28 degrees Celsius, so we will have to incubate it at 28 degrees Celsius, 200 RPM overnight. And similarly, we have to inoculate from the primary culture to secondary culture, that will be of 100 ml LB media, and again it will have all the antibiotics which specific to that. And after that we will just simply co-cultivate our explants, which I have shown you earlier. We will just simply co-cultivate our explant, and the secondary culture which I have showed in earlier slide, we will take the secondary culture and we will just centrifuge it and obtain the pellet, and the bacterial pellet will be used to adjust and we will adjust its OD between 0.8 to 1, and we can do that by diluting it with our liquid MS solution, or MS without sucrose and agar. For that case we have used it. And after that, the in vitro grown plants, as we have to sterile the plants only once when we are setting up our plant in the tissue culture facility, and after that we can simply take the in vitro grown plants and we can use it directly after excising, and we have to place it in a falcon tube with the mixture of this, our bacterial or agrobacterium, which is harboring that vector and containing the guide RNA specific for different genes, like I have talked STBHLH47 and ST16 dogs. So after this we have to incubate it for 30 minutes, and this all part you have to standardize. Like this is not, you can just follow exact like this. You have to optimize it for your plant. Different plants have accordingly you have to adjust. So after that, after the co-cultivation, the next step will be to eliminate the excess amount of agrobacterium, as we don't need the agrobacterium now, because it will lead to contamination and we don't want any contamination, as it will hinder our plant growth. So for eliminating of this agrobacterium strain, we have to simply transfer our explants in the MS medium, and we will supplement it with a broad spectrum antibiotic, and in this case we have used timentin. You can also use cefotaxime. Both of them are broad spectrum antibiotic, and the concentration we have used here is 250 mg per liter. So after that you have to incubate. After washing with this medium, you can simply incubate your explants to suppress this suppress agrobacterium. And after you have successfully eliminated the excess agrobacterium, you simply have to look for the successful transformant events that you get, as the transformation frequency is not that high that you will obtain each plant to be transformant. So you have to select them, and here the selectable marker is hygromycin for plant. So that's why here the MS medium contains hygromycin as well, and we have also included that broad spectrum antibiotic to control the excess agrobacterium. So after that, the transformed explants will be cultured in the regulated environments with controlled light and controlled temperature, and after that the lines which will survive will be our transformed explants, and the line which appear to be yellow are untransformed lines, and they will eventually die. So like it in case of potato, it takes roughly around 1 month for us to get transformed explants, but you will, if you do it regularly, you can uh, say that like within 2 weeks you are able to distinguish between the transformant and untransformed plants. But to get a more clear view, you have to wait for like a month, I would say, in case of potato. So, and here I have shown the pictures. So in the pictures I have also included leaf as an explant source, and for the leaf I have used hairy root agrobacterium strain, that is agrobacterium rhizogenes. So as you can see that after some time you are seeing here hairy roots, and in case of this I have used the GV3101 strain, that is the agrobacterium tumefaciens strain, so that's why there are no hairy roots, and you can see that the untransformed plants are becoming yellow and start to die, and while the others are flourishing well. So uh, this is how your transformed and non-transformed plants will look after some time. Like in our case, it took us around 1 month. So after selection of the transformed plants, the next step is the regeneration part as well. So like in the previous slide, I have shown you by using, like you can use leaf as an explant as well. And at that in that case, I have used the strain A4, that is the Agrobacterium rhizogene strain. So that's why you are seeing hairy root there. So if you are using leaf as an explant source, you have to optimize this regeneration of the transformants as well, because usually you don't get a whole plant out of this leaf, not explant but leaf. But if you are using internode as a explant source, it is more easy for that. But by using this rhizogenes, Agrobacterium rhizogenes, there are certain advantages as well. So that's why we have optimized transformation using both of these strains, like Agrobacterium rhizogenes and Agrobacterium tumefaciens as well. So for the transformation, you have to like, we don't keep our plants too long for sitting on a single media. We regularly subculture it, like in 10 days, within an interval of 10 days. So we transform them, and you have to optimize the ratio for auxin to cytokinin, or in the reverse order, depending on whether you want to initiate the shooting or rooting. So in tissue culture, we initially focus on shooting, and after shooting, after the shoots emerge, we follow, we transfer the media on rooting medium. So the shooting is first if you are using callus, like so. Initially, you have to wait for the shooting, and after that, you can transfer it for the rooting. So the successful regenerated plants will be further used to analyze for the mutants, because we have till now using all these things, we can say that we can successfully develop transformed plants that is harboring our vector specific for that guide RNA. So we'll have to analyze the mutations in the developed lines. So for that, here I have shown that in case of potato, using that Agrobacterium rhizogene strain, I have transformed the leaf, and I have shown you in the previous slides that the leaf was having these hairy roots, and the hairy roots were further grown to generate this this callus, and which was further transferred on the shooting medium. And here you can see that shoot has emerged from this callus, and after that, uh, once the shooting has been done, we don't need to transfer it again on rooting medium, as it already is having so many roots. So we don't need rooting medium for this particular setup. And after that, the plant was further grown, and after that it was acclimatized. So this is the complete schematic representation, and here I have shown you two different calais, like these are two different lines in my case. So again, so till now, this was the whole thing which I have tried to illustrate you and outlined you with the help of these slides. So initially, our first step is to sterilize. So like our plant, suppose, is growing in a field or in a pot. So we'll take the explant and we'll wash it or surface sterilize it, and after that we'll cultivate it in this medium, MS medium, or simply if we have a in vitro grown plant, we can simply take the explant from here, and after that we can co-cultivate it, as I have shown you some pictures as well. For that, you have to have your construct inside that agrobacterium, and that culture will be used for co-cultivating here, and after that we'll co-cultivate it and wash it to eliminate any excess agrobacterium. And after that, we can regenerate a plant if we are talking about using this hairy root system. And after that, the generated lines will be used for DNA isolation and cloning and further detection of mutations. So this is the whole pipeline, I would say, to follow when you are designing your own experiment. So And once you have isolated DNA from your transgenic lines, from your mutated transgenic line, you have to confirm it. So this picture is, shows the confirmation part. So here we have three different gel images. One is showing the one is showing our role B gene, primer specific to role B gene, and the second gel is showing our in which we have used this HPT2 gene, which is present as in our vector. Like this is resistance for hygromycin. So we have designed primers specific to this HPT2 role B, and in the third gel image we have used Cas9 gene and designed the primers accordingly, and we have used the PCR amplification, and here in the lines, in the wells, we have obtained the band. These are all transgenic lines. So eventually we came to know that these lines are confirmed positive lines, and our vector is inside those lines, or I would say our tDNA region is integrated in the chromosome as well, as because we have also used role B here for the detection. So, and you can also see that some of the wells are empty as well. So it's not obvious that you will get all the lines positive. You will also get certain lines negative as well. So you have to screen. This is the screening part basically. After you have generated the transgenic lines, you have to screen it. So this is the screening part. So you have to select all the positive lines, and you have to only use those positive lines to further detect mutations, for which what we have you have to do. Like if you have, suppose, gene of length, suppose earlier we have taken 1500 base pair, that PIF gene was, our candidate gene was 1500 base pair in our previous tutorial. So like suppose your target gene is 1500 base pair long. So for the screening part, you will not amplify it from the start to the end. You will just design some short primers just for like in a range of 200 to 300 base pair region. In between your target region should lie. As we are not amplifying the full stretch for the confirmation part, because if the stretch is too big, there may be some sequencing errors as well. So once you will get into it, you will came to know that using a small part for the screening is good. Like you don't have to fully amplify your gene in that case. So we will design specific primers for the region. I have told like 200 to 300 base pair, and our target region should lie in between them, and we'll simply amplify it, and we will similarly get bands like this depending on our gene. And after that we have to elute it and simply clone it in cloning vector like PJET. You can use PGMT, PJET as well. That all depends on your lab and whatever you want to use. And after that, after cloning, you have to simply send your samples for sequencing, or if you have access to any sequencer, you can use yourself sequence your samples, and the sequencing results will look like this. Earlier, in the earlier lectures, Kashmir sir has also told you about these peaks that you get after sequencing, and if anybody remembers what he told about how the peak should be, like the more, it should be like crisp, it should not be, it should be sharp basically, it should not be a blunt or a broader peak. So this defines the quality of our sequencing result, and based on this you can align your sequence, your different sequences, and here this top sequence is the wild type sequence, and these are our lines which I have numbered 2, 7, 5, 6. So after you align it, you will come to get this whether something is missing from your target region is not target region or not. So this is our target region, and I have highlighted our PAM, that is TGG here. So this is how you basically screen. Like the more number of mutants you have, the more you have to screen. You have to amplify that particular stretch of your gene and clone it and further sequence it. And sometimes one-time sequencing is not enough to be sure about it. Like in our case, potato, as I have mentioned earlier, that potato is tetraploid crop. So there are 4 copies. So it's not that easy to get these mutations like in a single sequencing result. So you have to clone it multiple times, and you have to check whether you have any mutations or not. So this was the selection part. So to conclude that using this described methods, we were able to optimize CRISPR-Cas9 system for potato, and we have successfully generated transformant and regenerated them using these mentioned protocols, and the positive transformants were screened for mutations as I have showed in the previous slide, and we were successfully be able to knock down this, for which we have analyzed it for like metabolic profiling, and we did HPLC ICP-MS as well. So we have conducted various other experiments to support the successful knockdown of both of these genes. So you have to simply follow all these things if you want to design your experiment, and if you want to read more or get an in-depth literature on how to do these things, you can follow just these papers if you are working on potato. Otherwise you can select from a lot of papers are available on Google Scholar as well, but if you are working on potato, you can follow these papers as well. These are our own publications. So this was it, and I hope I can clear any doubts or questions you have. The slides were very basic actually. I haven't made them that.
About like we would categorize the explants into like if they are growing well and or if they are
basically in transformants and non-transformants.
Yeah.
Yes.
So like they could be some other reasons of like not growing despite of being non-transformed.
But all the explants are growing in a similar kind of growth environment. The media is same. So I think the ones which are not growing are certainly the transformed plants, and ultimately we are also making it sure by PCR amplification as well. So I think any other questions, or from the previous tutorial as well, you can ask anything if you want to. Yes.
From previous tutorial, like designing vectors first and then making them express in the cells. So how do we control the expression of those genes like Cas9 and sgRNA inside the cell? Do they keep on expressing all the time, or
No, that follows the cell's own mechanism for its. We just simply provide them a good promoter, or if we want to specifically target some tissue or some particular area, we will put that tissue specific promoter in the upstream region of our
because
that.
if it keeps on expressing all the time, the amount of sgRNA and Cas9 will increase and it may lead to
Off targets.
off targets.
Okay, that may be one thing, but it usually don't happen like this, because the organism, maybe you are working, I think you are working on humans, I think. So there are 2 alleles, I think. So it wouldn't cause that much of a, because the system has another copy of that particular gene as well. So to obtain a homozygous mutation, you have to, like in case of plants, you have to cross it and get the homozygous mutation. So
Can we directly synthesize sgRNA and make nucleoproteins? And
yes, we can do that. For that, you don't have to need any vectors. Like you need vectors for in vitro transcription, and once you have in vitro transcribed your single guide RNA, you can just simply purchase or express Cas9 protein, And if you can purchase, there are several options available from Thermo, I think. You can directly purchase this Cas9 and mix them with your in vitro transcribed single guide RNA, and you can mix it in a specific particular ratio that depends on what organism you are working on. You have to find it in literature, and you just have to simply mix it and transfect it with biolistics or any other suitable transformation method that you can use.
Do we have to amplify sequence the gene multiple times because it's tetraploid or like
No, in diploid, yes, you have to do it in multiple of number of times, because it's not like that you just amplified it and sequenced it one time and you will get mutations. It doesn't work like this. So because there are multiple things that happen in PCR as well, there are multiple reads, multiple amplicons, and if you run the PCR too long, the amplicons will be more of the non-mutant variant. Like you are getting what I'm trying to say, that there are two alleles, like if we are talking about, there are two copies basically. So one is the normal one and one is the mutated one. There may be some case when both of them are mutated, but that's very rare. So if one copy is normal and one is mutated, and if you are in a PCR cycle, PCR reaction, you are keeping your reaction for too long, then there are chances that you will not get the mutations in your sequencing result, because in sequencing it will pick up the template that is more readily available, and the non-mutant one will be readily available. So I think these were some of the issues that we face, but now we know that this happened. So you have to have an excess of sequencer. I think that would be very good. Otherwise, you can also outsource sequencing, but to have access to a sequencer is an advantage, I would say. Even we also don't have a sequencer here, so we also outsource. So, any other questions? Okay, thank you.